2021. I want to thank you all for coming this quarter, and I especially want to thank all of you who are joining virtually from other institutes. I hope you've found the lectures this quarter stimulating. My task today is to introduce someone who needs no introduction. Of course, I'm talking about Professor Vicki Meadows, the director of our astrobiology program here. She came to the UW in 2007 after many years at the Jet Propulsion Laboratory. Throughout her career, she's amassed so many awards and accolades, it's almost impossible to keep track of them all. A few of them that I'll mention that are highlights include being the PI of the Virtual Planetary Lab for 20 years and counting. And that includes now being a co-lead of NASA's Nexus for Exoplanet and Solar System Science, Nexus. She's one of the co-leads of that, which is a research coordination network that connects a lot of different people that are working on planetary science and evolution of planets, both here and around other stars. She also won the Frank Drake Medal, I believe in 2017, thereabouts. She has served as the director of the NASA Exoplanet Analysis Group for several years. And just this year, she was inducted into the Washington State Academy of Sciences. So congratulations on that again, Vicki. On top of all of that, amongst her many leadership roles, she was the co-lead for our co-chair of NASA working group this summer on standards of evidence for biosignature detection. And she's graciously agreed to give us the top-level executive summary of that today, and maybe it'll even evolve into some discussion today as well. So with that, I'm going to pass it over to you.
Okay,
please. Thank
you very much, Rory, for that lovely introduction. Yeah, I think of them, most of them are not really awards, they're more like responsibilities. And so that's what I've been taking on. So speaking of responsibilities, this is a bit like, you know, you come back in autumn quarter and it's like, what did I do with my summer? Well, this is kind of what did I do with my year? So this is going to be a summary of an effort that we actually started back in February of this year to put together a community workshop to start tackling some really difficult questions about, you know, what does it mean to have robustly detected a biosignature, and how do we convey what we know and what we don't know? In doing this, I was helped by a fantastic array of people, including my partner in crime, Heather Graham, who is wonderful and without whom I would not have been able to do this. So she is representative of Enfold, which is the Network for Life Detection. It's a NASA RCN like Nexus. I'm going to talk a very tiny bit about Nexus and Enfold just to get people up to speed on what those things are. And of course, we had this fabulous scientific organizing committee, which was about, I think, 24 people, at least initially. And so you can see them up there. And this workshop was also run in 3 different time zones in the US and also in the Asia Pacific region and in Europe. And so we had some fabulous chairs that you can see listed there for people who helped to get that workshop running in all of these different zones. So today I am going to talk about, okay, it's not letting me do that. All right, for some reason I've lost control of my slides. Okay, that will work. All right, so in this presentation, I'm going to address, you know, why did we have this workshop? What happened at the workshop? Who was involved? What was the outcome, and what still needs to be done? And I've given presentations like this to a bunch of sort of oversight bodies, and the response has been very interesting. Some have really focused on the science of what we did, and others have focused on the actual mechanism of what we did. And so I'll talk a little bit about both. Okay, so why do we need to have a workshop on biosignature standards of evidence? So the motivation for this workshop really was the realization, and this was highlighted in a paper that was just recently published in Nature by Jim Green. Took a while to get published, so we were aware of this paper before it actually was published. But it's the realization that, you know, the detection of extraterrestrial life, you know, in our solar system and beyond will likely be neither instantaneous nor unambiguous. But on the other hand, it's an extremely high stakes scientific achievement with high public interest. And so given that we have essentially something that's going to require multiple measurements, potentially a large fraction of the community, and yet may end up not being, you know, very definitive, you know, how do we go through the process of getting a more robust result and being able to communicate where we are in that process? We also realize that extraterrestrial life detection is now an explicit goal for both planetary science, and you've seen with the recent release of the Astro 2020 Decadal, also even for astronomy and astrophysics. The top goal there now, the one that is driving the development of the flagship mission, is understanding worlds and suns in context. And the end goal of that is, in fact, to search for biosignatures on extrasolar planets. So clearly, you know, this goal of life detection is becoming extremely important for a lot of NASA missions across many NASA directorates. There also needs to be an effective way to convey the fact that this process is multi-step, that it's a continuum of things that we may do, and it may span multiple missions and measurements. I think, you know, some people are concerned that, you know, if your mission doesn't, you know, unequivocally detect a biosignature, then it's somehow failed. And we don't want people to be thinking that. We want them to see, oh, here we are on the arc, the path towards definitive life detection. And this is what this mission does on that path. And there will be other missions subsequently that will help us with this particular process. And then the timeliness is that, you know, with increasing claims of possible life detection coming up here, it's really important to understand the best process we might have for vetting these claims, and also how to report and convey that information to the public and even to our colleagues when it can be inherently complex and quite interdisciplinary. So Heather actually put together this timeline of sort of major claims of life detection. You know, we had one every decade or so for a while, and then we've gotten quite a lot very recently coming out. So acknowledging that it's important and potentially also timely to be able to plan and execute a rigorous multi-step process for life detection and to be able to convey accurate levels of uncertainty as we go along this process, it would be very helpful to have some sort of progressive framework that values and contextualizes this series of results and says, even though your result is only step one on this process, we love you doing that step, right? You have to value each of the steps along the way towards a more definitive detection. Community guidelines, so not rules, but guidelines for claims of biosignature detection that would sort of set up the path and also enable more efficient scientific assessment would be valuable, and to ensure that promoted results are as robust as possible. And then community consensus guidelines for how we should clearly communicate our results to each other and to the public, how we talk about the significance of what we found and what we still need to find is also really, really important. So as I said, there was this paper written by Jim Green and colleagues, and Jim is a senior scientist at NASA. He also felt that we needed this kind of a framework. And so there was this Nature paper put together, which was supposed to come out actually before the workshop, but due to the vagaries of Nature publishing, ended up coming out, I think, on the second day of the workshop or something like that. But it was only finally published actually last month, but it came out sort of a pre-archive version as well. So that paper was really a call to action to the community. You know, Jim wasn't going to solve our problem. He was just going to say, hey, this is an issue. It's something that the community really needs to address. And so the community response to that was to develop this Biosignature Standards of Evidence workshop. So the Standards of Evidence workshop was put together by both the Nexus and Enfold research coordination networks. So I'll just explain what those are, because probably some of you heard them flying around. And it's relevant to you here because anybody who's in the Virtual Planetary Laboratory here at the University of Washington is also a member of Nexus. And so I'll explain what that is. So the Nexus, the Network for Exoplanet System Science, is an interdisciplinary research coordination network. It's dedicated to the study of planetary habitability and to the search for life on exoplanets. So it's really quite astrobiology focused. So that's one thing you should remember there. We would like it to be, even though it's very much, all of astrobiology sits in planetary science at NASA, but also has, you know, when we talk about exoplanets, extreme relevance to the astrophysics division, and also potentially to earth science and heliophysics, which are the other two science directorates at NASA. So the hope is that Nexus will be cross-divisional, bringing scientists from these different disciplines together to look at, you know, system science, looking at exoplanets as systems to understand how we might tell if they're habitable or have life on them. And it's a way for NASA to really, and here's that word, leverage investments in research and missions to essentially, you know, get us working as a much larger interdisciplinary community to help support these missions. It's one of the 5 research coordination networks that incorporate ICAR teams. What's an ICAR team? An ICAR team, I think it's the Interdisciplinary Consortium for Astrobiology Research. That is what the NAI became essentially as far as funding mechanisms are concerned. So if you were going to get something like an NAI group, which is what VPL started out as, then you would the next thing we would have to do is potentially go to become an ICAR team if we wanted to do that. And then the ICAR teams also are incorporated within the RCNs. But the RCNs themselves also include single PI investigators and so on. So the Nexus is very broad. It has about 410 members and 66 teams within it. Again, some bigger ICAR teams, some mid-sized teams, some smaller teams, and spans, you know, international, an international community. Okay, so for Enfold, I don't have as good a slide for Enfold. I just took a snapshot from their website, but Enfold is the Network for Life Detection. So it's another research coordination network, which, as you can imagine, has principal goals of advancing life detection strategies and capability. So that's essentially what they're there for, to catalyze collaboration amongst the community and thinking about this, to support NASA programs and missions, and to foster community development of ideas about life detection. So Nexus and Enfold got together and put together this particular workshop. So our goals included, as we justified, you know, developing that generalized framework and process, increasing confidence in and communicating life detection discoveries. We divided the workshop into two parts, one larger than the other, but looking at what we might need as far as standards of evidence or a framework for biosignature assessment, and then looking to begin the discussion about what we might want for a reporting protocol when it comes to life detection. The goal was to sort of document any community consensus along the way, and to try and develop something that was applicable to everybody. And so that was our biggest challenge, was to try and figure out, is there a process that will work for someone looking for stromatolites, someone looking for organics on Mars, and someone looking for life in an extrasolar planet atmosphere? We wanted it to be efficient for the community to implement. We didn't want to have to go easy step, really hard step, back to some easy steps, right? We wanted to make sure that as we progressed through any assessment framework, we were doing like the next hardest step as we went. Again, it had to frame the search for life as a continuum of objectives rather than my mission will do this one thing and it will be the answer. And that it was really important that it helps to convey not only what we know, but also what we don't know and what needs to come next. So, sorry, very wordy slides. So the workshop development, we started by outlining, you know, begin with the end in mind, one of those 7 habits of highly effective people or something. So we started by outlining the needed sections of the workshop white paper that we hope would come out of the workshop. We then worked backwards from that to develop a workshop that was in flipped format, which you here will probably know very well. So what we did was we put together pre-meeting materials that would have taken the place of, say, plenaries, and were available to all the meeting participants in advance. And then we flipped it so that when we got together on Zoom, we did basically nothing but discussion and reporting out. So the workshop was only 15 hours long, of which 13.5 was actual active discussion and not breaks. So keep that in mind. So I talk about what the outcomes are, that we really just had about 13.5 hours to try and put all of that stuff together in the initial discussions. As I said, we span 3 different time zones. Because I don't know about you, but I am very tired of attending European conferences at 3 a.m. in the morning. So we said, okay, let's be kind to everybody. Let's try and set something up so that it's actually within the normal business hours. And so to make significant progress in that 13.5 hours, the co-leads and the science organizing committee put together sort of a straw man framework of how we thought biosignature assessment might go. And we let the community kind of try and tear that to pieces. In fact, we actively encourage them to try and break the framework with their specific examples. And so that's what we did to sort of get the process going. This was the white paper outline. We wanted to describe the motivation, describe our unified framework if we could come up with that, give worked examples for different fields of how the framework might apply to their particular measurements of biosignatures. In the end, we wanted to have further applications, but a lot of the discussion then centered around how the framework might affect mission life cycle for missions that look for life. And so that became, in the end, a section of its own. We wanted to discuss reporting protocols and then findings and future high priority research avenues. And again, I just want to stress that this was the beginning of what will likely be quite a long process. So it's important to understand where we started here. I imagine there are going to be future workshops that go into sort of more detail in some of these aspects. So I'll skip over this a little bit, but working with the SOC, we invited a bunch of participants, sent out the general call, advertised it to everybody. We ended up getting 355 applications for 80 participant slots. So I just want to say that this was super, super exciting to a bunch of people. We, in the end, selected about 82 people who would engage in the active discussion. And for those who weren't selected for the active discussion, actually quite a lot self-identified as people who didn't want to interact, but who wanted to come and just learn and to listen in. And so for them, we developed a second tier of participation, which was being able to listen to everything that was being said and to be able to give feedback in Slack. We had a workshop Slack as well. So in the end, we had sort of probably over about 160 people actually actively involved in the Slack. So we provided reading materials and we provided videos and so on for people to get up to speed. And if you're interested, the reading materials list is up on the Nexus website. And these pre-meeting videos are just going to be released today, actually. And so they're quite, I think, good for instructions. We tried to keep them to about 25 minutes or less. But we had a bunch of things that were just sort of an introduction from me and Heather and the charge, overviews of state of the field by Dave Demaray and our very own Sean Dummel-Goldman, if any of you are old enough to remember Sean as a postdoc here. We had worked examples on various previous life detection claims, so things like the Viking biology and ALH 84001, and of course, phosphine on Venus. Jill Tarter very graciously agreed to give us a fabulously titled video on when do we drink the champagne, about sort of the steps that would be needed, for example, for extraterrestrial life detection a process so that we could learn from them. And then we have a bunch of instructional videos, including from our own Dr. Jevon West from UW, for the Calling Bullshit author, who these people, gave us some overviews on sort of key techniques or issues that it would be nice to understand as we have this discussion. So the workshop ended up with 125 active discussion participants. Oh yeah, so 215 asynchronous participants, 40 of which signed up for Slack. And basically we had about, and this was a surprise to some people, we had 25% exoplanet observers, but the vast bulk of the people who came to this workshop, 57%, were solar system and planetary science astrobiologists. So that really just reflects sort of the composition of our community. And 18% early Earth and paleobiology people as well. And we actually split the participant demographics with about 50% pre-tenure and 50% post-tenure. So we had a fairly large early career component in this as well. Okay, so we documented all of our discussion outcomes. So we set it up so everybody had, every breakout group. So we had sort of discussion topics. We had breakout groups. Everybody went into breakout groups, had a huge discussion, then wrote everything down in notes, and then wrote a summary presentation, which was presented out in the report. We ended up with 379 pages of notes. So there's a lot in there. Everybody was sort of very, very, gung-ho to document what they were, in fact, writing about. And so we were able to use those notes to put together a white paper. The workshop itself, basically, this is the way it was structured. So first of all, we put out that straw man framework and we said to everybody, okay, break it. So we gave it to people in their specific disciplines. So if you were, for example, somebody who looked for stromatolite biosignatures in the early Earth, you were in a group with other people who did the same. So the first discussion was, okay, does this framework actually work for you guys? So with your specific biosignature target and your specific technique, does this in fact work for you? And so they did that first to see whether they could, struggle with it and see whether it worked or if it just didn't work for their particular thing. And then we homogenized everybody. We broke everybody up into random groups. And we asked them, okay, now talk across the disciplines and see whether you guys have any common problems with this or whether there's ways that we can actually reconcile what's going on. And then in day 2, we had identified problems with the framework and we worked through them all in the morning. Day 3, we actually sat down and started trying to write the white paper. And day 4, we started the discussion on the reporting protocols. And of course, we couldn't get to a whole reporting protocol in the 1 hour we had to do that. But there was an amazing amount of creativity across the sort of 8 to 10 discussion groups that we had. And we documented that and a bunch of ideas that people had for that. And the continued engagement was pretty high. We had about 84% overall for 4 days in the US group and about 90% participation continuing for the 3 in the satellites. Whoops, sorry. I actually don't know how to go back. You're back there. Okay. Thanks. Okay. Yeah, can you actually help with that? Oh, this one, okay. Yeah. Here we go.
One you missed.
Okay. All right. So our goal was, the principal goal was to try and get to this unified framework for biosignature assessment, which I promise you I'm getting to in just a little bit. So we tried to, develop a series of steps that you could go through to try and assess a biosignature. and that it should be sufficiently general that it could be applied to multiple planetary environments, measurement techniques, and biosignature targets. And so these were the questions we gave everybody, which was, first try to break the assessment framework, try to map how you would use it, and then assess whether or not it actually works. And then in a diverse group, design a framework that actually works for multiple people. Again, problem solving, outlining, and then finally discussing that reporting protocol. And in the reporting protocol, we asked the question, dream up an ideal reporting protocol for a claim of life detection. How would you like to see it actually go? And then discuss the incentives and disincentives that mean that you don't actually get that ideal process. And write those down. We also had white paper writing activities. And this was astonishing to me and Heather. At the end of the workshop, we had ongoing writing with the workshop participants. It was entirely optional, and about 60% of the attendees, so 74 people, agreed to keep working on this and to write the white paper, which was, an embarrassment of riches, really. It was fantastic, but we were actually unexpected to see that level of engagement in this. So we drafted the white paper with that group, and we essentially had a bunch of telecons that just were ongoing for a week or so. After that, people really wanted to discuss this particular topic. And so we ended up putting together a draft, which went out for public comments. Hopefully some of you saw that. I did send announcements around, and you were able to hopefully endorse it if you liked what we had actually written there. And so over about 100 members of the broader community have left comments as well, which we are in the process of working into the document. And it says that white paper will be finalized in early December. It's probably likely to be mid-December, given the response that we've gotten from everybody. Okay, enough of that. So the Biosignature Assessment Framework, what did we actually come up with? And this is the final wording after we tried to break it, and people, I think a large fraction of what the community really had to grapple with was like, what does any of this really mean? And are we being very clear in how we're describing things? So the first thing we said was, okay, this Biosignature Assessment Framework is going to have 5 fundamental questions that you need to answer to progressively be more confident that you're actually looking at life. So first of all, have you detected an authentic signal? Have you actually detected something that is in the environment and is not just some kind of artifact in your instrument, right? Have you actually got data rather than noise? Then have you adequately identified that signal? Is that thing that you think you've measured actually what you think it is? Or is it something else? Is there some contamination in the instrument? Is there something else in the environment that's confusing you? Do you have coalition of lines in a in a chromatograph, or do you have blended lines in a spectrum, that sort of thing? So do you have a real signal? Have you identified it correctly? And then you have to ask yourself the question, are there abiotic sources for your detection? Can you explain this without life? Go through that process first. Or maybe in tandem with the hypothesis that, is it likely that life would produce the expression in this environment? So people definitely did want to move away from, just thinking about life as kind of the hypothesis of last resort. And then finally, because we're scientists, given your hypotheses, maybe you've developed a hypothesis that says, no, I think this is an abiotic phenomenon. Can you make a follow-up experiment that tests that hypothesis as to whether or not that's actually true? And we developed this framework, and I'll describe it in more detail for the rest of the talk. But we ultimately decided that we would need to try and assess in some way our confidence in each of these steps. And we don't mean confidence actually so much in a statistical sense. Actually, the statisticians, basically let us know that we were not allowed to use confidence in that way. And I was like, yeah, but I'm thinking of like human, just everyday confidence. Like, am I confident that this is in fact correct? But anyway, we need to somehow assess in some way, how certain we are that we've met the requirements for a given step before we kind of move on. And then the workshop goal, of course, was assessing and challenging that framework with a range of examples. So here we go towards a generalized framework. So the 5 scientific questions that I just put up there were seen to be sort of, divided into 2 different types of things. Questions 1 and 2 were really about your biosignature detection. Do you have a real signal? Is it what you think it is? And then questions 3, 4, and 5 were more about assessment and interpretation of that signal. Is it due to life? Is it due to non-life? Can I prove it? And so that was kind of the structure. We emphasize that the framework didn't have to go in a linear way. And this is actually a diagram that Mike Wong put together. I think that Lisa refined, just to say that, you know, some of that detection and then contamination issues, it turns out in fields other than mine, you know, when we all got together and talked about these sorts of things, that, you know, sometimes one measurement would serve both purposes in a given field, or, you know, so it wasn't easy to just break things up into 1 then 2. It was more like 1 and 2 at the same time. And so we talked about the fact that even though the questions were linear, we might not apply them all linearly. But what we did decide is that it's probably a good idea to get a pretty robust detection before you move on to the assessment and interpretation phase. So let's get 1 and 2 nailed down. And then, you know, if we really are confident that we've got a real signal and we've identified it correctly, then we move into sort of level 2. And there was an acknowledgement. You'll see these little clipped out pieces of paper here at the bottom. These are just direct quotes from the notes from the workshop. You know, people realizing the energy between level 1 and 2 is high, that, you know, the amount of time and effort and the fraction of the community involved in verifying the signal is much smaller than it is in trying to assess the signal. And so again, you know, we need to decide on some kind of threshold where we move on to that. And as we move down in the framework, we're going from strongly disciplinary to interdisciplinary. So more of the community again gets involved. And people said, you know, remote sensing and in situ measurement should be on the same scale of how confident we are, even if they have very different conferences overall. So there was also a lot of grappling with how do we do this when we're doing multiple techniques? How do we get that together? And then at the end of the the end of the second session on the first day, we all started to write down, okay, what are the problems with the framework? How do we address these? So these were, the community just wrote down, you know, key things that people thought about. You know, is habitability guiding the search? Should we think about habitability first before we go into any of this stuff? Yeah, how well do we want to define 3 to 5? Yeah, do we have to use it in a linear fashion, or how do we use it? How do we incorporate preservation of signals, for example, into this? That was a big issue that came up with the in situ community. So we put down all of those problems, and then the next morning we all picked a problem and we went into a breakout session and we tried to deal with it. And so I'm going to just go through sort of in detail what each question means. One of the really big things that came out of this was that you know, the community really wanted to come towards a generalized framework, but a large part of that was just trying to understand each other's terminology and grappling with, you know, how they do things in a given field versus how we do it in another. And just, so it was a fabulous experience to just learn about all of that from all of the different fields as well. So our first question, have you detected an authentic signal? So in other words, do you have a real statistically significant signal as opposed to noise? And have you ruled out artifacts from the measurement, pre-processing, or analysis that might mimic a signal? Have you basically been fooled by noise? You know, again, have you accounted for all of the instrumental artifacts in this particular case? And is the result repeatable or reproducible or verified by other groups or supporting measurements? That's how you would get to a much more robust answer to this particular question. Yeah, so reproducibility came up quite a lot. And again, you know, when we do our science, we all understand that one of the major tenets of science is that it should be reproducible, right? But we don't necessarily actually do that in our science. And I think the community was pushing forward and saying, you know, for these types of claims, for saying I found life on Mars, maybe we should, you know, try and, you know, as a guideline say, you know, maybe our results actually should really be reproducible. Question 2, have you adequately identified the signal? You know, have you adequately ruled out other potential sources? So not noise, but other real things like contamination in your instrument or environment or other phenomena that could in fact produce the signal. And we had specific examples there on, you know, how we might get fooled in different fields about different things. And, you know, this sort of, again, from the community, little notes about how life detection missions should have, you know, collaborative measurements that complement each other so that we can, as we plan our missions, get a more robust result from a given mission. And of course, that's very, very difficult for those of us who work in exoplanet science, because you can have any data you want as long as it's a spectrum. So yes, we talked about sort of the luxury that some of these other fields had and potentially having multiple measurements and different measurements overall. So are there abiotic sources for your detection? So is it likely that there's a current or past process that might actually mimic what you're looking for? And then, you know, do you really understand the environmental context well enough to be able to do that and to rule it out? And there's this wonderful note from one of the notes, which is just like, oh, context is everything. We really, really need to acknowledge that we need to look at context. People called for multiple lines of evidence and a more expansive sort of exploration of possible abiotic mimics that might occur. So that was a big thing. For the people who work in situ, one of the big questions was, what does abiotic actually look like? Since microbes have contaminated everything, do we need to go to the laboratory to get abiotic processes that are truly abiotic? And then question 4 is kind of this parallel hypothesis. One hypothesis says that, maybe this phenomenon is due to life, and the other one, one is not due to life, and the other one says, is this due to life? How do you actually test that? And the idea that the life hypothesis has to be tested just as rigorously as the non-life hypothesis, and shouldn't just be, I don't understand it, therefore it must be life. So, and again, here's where environmental context really comes in trying to figure out whether life would actually plausibly produce something in this environment, and in fact, that biosignature might in fact persist in that environment if produced by life. And then further testing of the life and abiotic hypothesis, really just question 5 is kind of, okay, you've been testing 2 hypotheses here. Can you take us the next step forward in designing another experiment that will tell us about them? There are any quotes I want to pull out here. Yeah, again, multiple lines of evidence was really pulled out as kind of a central theme for our community. So kind of key themes that came out of this framework discussion that were very interesting. And I think this is another one of Mike Wong's. Mike Wong really illustrated this workshop for us. And this was in the notes. So they put this together in their breakout session. Biosignatures, for the in situ researchers, and maybe many of you who are in situ will resonate with this. But if you find a molecule or a molecule fragment, how do you tell it's a fragment of a more complex molecule? How do you tell whether it's not just a prebiotic molecule that has to be put together? Is there some sort of protobiotic system? Where are we on this spectrum of molecules and what they mean? And again, preservation also gets tied up into that. It's whether your molecule breaks up or is in fact available. So again, people identified a potential gap in trying to understand this kind of spectrum of prebiotic, biotic, and abiotic phenomena or molecules in this particular case, and trying to understand how we would actually discriminate between those. And then the acknowledgement you might have multiple working hypotheses at once, that you might not necessarily just sequentially test, is it not life? And then is it life? But you might be trying to do all of that simultaneously. So key themes that sort of came out from the discussion of the framework was that this generalized framework should actually work for multiple different fields, albeit with customized specifics for different fields. So for example, my idea of what a high signal to noise detection is in my spectrum might be different to what it might be for someone who's observing stromatolites or looking for organics on Mars. Recognition that certain steps are going to be more challenging and potentially require different thresholds for robustness for remote sensing versus in situ and extinct versus extant. I still think we should hold everybody to the same standards, but we realize that it might be a little bit difficult. Definitely more study of that false positive or pre or protobiotic expressions was called out as something the community needs to do. People wanted to think about working preservation of the signal into the framework. So this is the potential for false negatives because things are being destroyed. A general sense in the community that these biggest science claims really deserve extra validation and more rigorous than normal peer review. And then the question was, how do we do that? How do we support that? How do we encourage that? We obviously can't lay down the law on that, but how do we, set up kind of best practices for doing that? That we would need multiple lines of evidence for robust detection, and then, to get the best result, that the field would need more transparency as far as open data sharing, which a lot of research, of course, is moving towards, and of course, this idea of reproducible results, which is an idealistic goal for science, but doesn't always get implemented. As we were working on the framework wording, there was this inherent tension between trying to say something simple that could be understood by both scientists and the public versus trying to get into more complexity to really make clear what it was that we were trying to say. And then also this sense that, the burden of proof that something is truly due to life should also fall on those making the claims of biosignature detection. And that was something that we as a community felt was an important thing to do. So example applications of the framework. People were struggling with this idea of, when do we actually apply this? Does it get applied before we make the measurement? Does it get applied as we make the discovery? And the answer is, of course, yes. You could apply it in a bunch of different stages of life detection. So you could use it to place a discovery on a spectrum of confidence. We're really only at level 1 here. We haven't quite gotten to, ruling out other things. You could convey your uncertainty once we get something more like a statistical framework in place. We could use it to help communicate what we know. And the example I would give is when I get called by reporters, when there's been a discovery or a claim of life detection or something, the reporter's first question will be, how excited should I be about this discovery? Because they don't know. And I mean, it's up to us to actually convey, the importance of, it's like, wow, this is amazing. These guys have validated this and they've done that and they've done this. And, there's been independent confirmation and blah, 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 blah. Is a very different discussion to, well, we're kind of at the very first stages here. We've only had a single measurement, right? So it allows us to sort of convey where we are in that. And then of course, determining whether a given measurement or mission would advance our life detection goals and getting into mission development. So here's some worked examples that we went through. So again, the first step was, here's a framework. How does that work for your particular science? And so the writing groups that came after, one group was team worked examples. And they were amazing. So we actually have 4 very advanced worked examples in the white paper if you want to go have a look at them. But the stromatolite group actually get a huge prize for their graphics, which were fabulous, in looking at sort of the initial questions and what they would do in their particular field to try and figure out whether or not they were in fact looking at an authentic signal. So, sorry. Yeah, so, local field mapping to figure out, I have something that looks like a stromatolite, does it actually reside in sedimentary rocks? Kind of, is it where I would expect it to be? Wider field mapping to get context, to try and understand if they've been fooled by something. Looking at potential abiotic sources, either, metamorphic or sedimentary. Could something else produce this other than it being a stromatolite? And then looking at, if the structure is actually congruent with the environment, if it makes sense where it actually is. And if this environment would support the appearance of these types of structures. And then of course, looking at independent lines of evidence in field and lab as well, to try and sort of, make other measurements to look at things like, isotopes or other kind of geochemical evidence for it being a life-based structure. Then we went from something like that to something like this. And this is from our own, you'll probably recognize at least a couple of these names. So Eddie Schwiedemann and Josh Christensen-Tutton were there, ably helped by Sunny Harmon. And they put together, just the sort of the classic. Again, we weren't really pushing the envelope here as far as which biosignatures to look at. We were pushing the framework. And so they said, okay, given the framework, does it work for oxygen on an extrasolar planet? And so they came up with this amazing diagram, which kind of looked at, okay, if I O2 is identified, what do I do next? For the detection, I need to get, at least signal to noise 5. I would maybe bump that higher with quibble about it. Evaluate systematics and processing areas, try and detect it with multiple epochs or instruments or, at a different wavelength or something like that. Identification, look at a different wavelength. Identify or exclude, look for other photochemical things like ozone that might verify your detection, et cetera, et cetera. And then, either it's disproven and you move on to your next target, or it's ambiguous or confirmed and you move on to level 2, which is really trying to assess what's going on here. And I think, kudos to that community. They already have a lot of previous work, which I think some of the in situ people were a little jealous of, previous work of false positives and trying to identify other ways that, the planet and star could potentially produce oxygen. So we have things that we could look for in the spectrum to try and rule all of that stuff out. And then our other 2 worked examples, Heather and colleagues put together one for in situ agnostic biosignatures. So not going in presupposing a metabolism, but just looking at things like the distribution of chirality, or looking at things like distribution of isotopes and trying to see if they could, if they could use the framework to try and get sort of a more robust detection by the framework. And we also had an example from Mars of looking for organics on Mars, which Dave Desmarais and other colleagues put together. So you can have a look at that one in the white paper. We talked about applications of the framework to the mission lifecycle as well. And that discussion really took off. I wasn't expecting that to be as quite as in-depth as it eventually happened, but Jen Eigenbrod and Sean Goldman both really got into that and wrote a pretty nice section of the white paper on as well. So, the point is that as you develop a spaceflight mission to look for life, the framework could be in the back of your mind as you develop a whole bunch of things, including this thing called the science traceability matrix, where you go from, what is my scientific goal? What is the objective, a breakdown of that? What is the measurement I need? And then do I have, what's the requirement on the instrument from that particular measurement? And so, each of these steps, question 1, confirmation of the detector's signal really requires planning and knowledge of calibration, making sure your instrument has the sensitivity required. Question 2 requires understanding any sources that might interpret the data and trying to proactively make sure that you guard against things like contamination or possible ambiguity in your identifications. And 3 and 4, those can be enhanced by pre-flight laboratory field or theoretical work to help you better understand the potential for false positives in the particular environment you're going to study. And also, helping to tell whether or not life is likely to produce that type of signal in that environment. And then, of course, question 5 places an emphasis on ensuring that the science traceability matrix and the instrument suite can actually get you a follow-on observation. And if they can't, that's okay. That's okay. Maybe that's left for the next mission. But at least you can define up front and say, look, this mission will actually take us, this way through the particular cycle. And then I think one of the most fun and possibly controversial parts of this particular workshop was sort of discussing a reporting protocol. Once we have a claim of life detection, how would we like that to be communicated to other scientists and to the public? And are there things that we can do, or are there things that we even need to change in the structure of our own community that might help to produce a better process? So we tasked that group with, imagining sort of standards for publication that could encourage the interdisciplinary collegiality and allow publications to actually accept open, incremental, or even negative data. At the moment, there's a disincentive to publish negative data. I went and looked for this biosignature, I didn't see it. It's not necessarily a paper that gets published. Although there was one recently on technosignatures, that was exactly that. So how do we incentivize the community also to adopt any sort of guidelines or best practices? How do we make that something that they would like to do? And what are the obstacles in preventing us from adopting these sorts of guidelines? And can we develop some sort of organized mechanism for claim validation? If you suddenly have a biosignature, where do you go? Do you go to your friends over here? Or, is there a directory that says, hey, I'm using this particular technique. Where's the other lab that could potentially try and verify this for me, for example? And then, of course, sort of discussing, how scientists can improve communications with one another as well as the public. And we also realized that we were just mostly scientists at this workshop. We did have representatives from publishing and from the media, but they were sort of one person each. And they were fabulous in the breakout sessions and giving us a lot of input, but they really were just one representative each. And so the acknowledgement that if we want to go further on a reporting protocol, we really have to get those stakeholders back in for a more rigorous discussion. So participants discussed at length, mechanisms that help improve collaboration. And for example, by funding and organizing pre-publication claims or verification of science activities, or even post-publication, but just, being able to incentivize people to actually do the reproducibility validation effort. So people talked about, potentially, here's some of the ideas that came from the community. We could maybe have a rapid response funding pool to support verification if someone came up with a life detection claim. We could do some sort of coordination of collaboration and data sharing, supported potentially through international networks or some kind of professional society, or even a new astrobiology society, which we don't have. We don't have an astrobiology society. Maybe develop sort of best practices where there's concurrent publication of discovery and verification, if that becomes a thing. And that it's okay to do stepwise reporting of results because again, it's just pointing out that it doesn't have to be a definitive answer. It can be a step along the way. New publication models that encourage interdisciplinarity of these incremental results or even negative data. And, try to partner with the journals and publications as well to try and prevent miscommunication of results as they come out. But again, if we can point to some kind of scale and say, we're here, it makes it easier to convey what's actually going on. And then of course, just when publishing our results and when communicating with the public, to be sure that we include and explain any error or uncertainty in our results, which is not something that always gets emphasized, but that's something that could be put out there to say, look, here's what we know it to this degree of certainty. Here's what we don't know. Here's what's hanging out there. Still needs to be done, and to just explain that this whole process is really part of a much longer scientific process overall. So this is basically my last slide. So obviously we only had 13.5 hours. We did what we could in that time. And I thought the community was amazing in coming together and pushing forward. The white paper is 60 pages long. We have all of these, ideas that are buzzing around. But there's clearly a lot of extra work that needs to be done. And so I've said 3 high priority areas, and then I wrote 5. So there's a whole bunch of priority areas for future research. Further development of these worked examples, other fields may be looking at whether the framework actually does work for them, or how it might actually shake out in reality. And including potentially a more detailed set of criteria that could work for field-specific guidelines. If I'm doing exoplanet remote sensing, what's the kind of process that we'd like to have? And again, that might help feed into mission requirements also. We need more collaborative discussions between biosignature scientists, data scientists, and statisticians to look at how we might incorporate a stepwise error and uncertainty and build a more quantitative scale for the hypothesis testing. Somebody put out the idea of testing Bayesian frameworks on biosignatures for early Earth. So if we develop these kinds of frameworks, there's already data sets with error and all sorts of other things, baked into it that could potentially be used to test these. Also looking at how we can incorporate other things that are out there in the community, like the Enfold, the CLD life detection knowledge base. So to try and see if there's other things that are existing in the community that could be used to help support the framework. And then of course, a more focused discussion on this reporting protocol that includes publishers and journalists and science communicators all together, trying to figure out, how do we best communicate both the joys and the traumas and the advances and the things that we still need to be done when we're looking at life detection discovery claims. So I'm going to stop there, and hopefully we have some time for questions, because yeah, I'd love to hear people's feedback and interest in this.
Thank you very much, Vicki. So we have time for questions. We'll start here in the room. And if for those of you who are online, feel free to type your questions in now and we'll get to those in a minute. So questions in the room first.
This is great, Vicki. It's great effort, but my vision is there won't be a checklist if you open bottle of champagne and you get so many checklists go to the positive. In the past history of planetary science, there's almost any topic like this, especially involving many disciplines, there are really hard-nosed discrepancies. Groups hate each other and they won't agree. It may take 30 years before there's some coalesce, but eventually, hopefully, the truth comes out. So, but this is a big deal with tens of billions of dollars going into this, and you really want to know, when you can open the bottle of champagne. And so I was wondering, what did the group think of like the top 5 or 10 ways that we might get the bottle of champagne in the next couple of decades?
I think the answer is there are multiple bottles of champagne.
Could you summarize?
Oh, sorry. I was trying to summarize. Okay, I will try to summarize this question. So John said that that this process can be a very long one and it can be contentious with multiple teams sort of, not agreeing. And so the whole process could be very long. And so the question is, when do we get to open the bottle of champagne? What are sort of the top ranked milestones that we would think, where champagne would be warranted? When do we get to open that bottle? And my response is that I think there are multiple steps along the way where we could potentially open that bottle. And that was kind of the point, was that this wasn't an on-off, we have life or we don't, that it really was this long graded process where we were trying to develop. So I think, yeah, every step along the way, personally for me, I think if you've actually got a real signal and it is what you think it is, definitely champagne at that point, right? You've got something you can interpret that's unusual and needs to be looked at. And then, you would have these other points along the way. But I think also what you're asking is in some sense a very field specific thing, and it goes to the specific worked examples of, when do we as a field think that, we've gotten to this particular process. But to your question about sort of animosity between groups and this long contentions and this long process, I think people are trying to obviously avoid that. And part of that would be to set up sort of best practices and norms, and to also have sort of open access to data. I know that some of these debates have gone on for a long time simply because the ability to very rapidly assess or verify results has not been available. And I would point to the phosphine on Venus debate that's going on right now. I see that as a very accelerated part of that process, and that was due in part to the fact that the discoverers made their data available immediately, and so people were able to sort of immediately assess those sorts of things. It didn't, I think you see a very rapid evolution of what's happening there. So there are processes and things that we could potentially do. And maybe I'll be a bit naive here too, and say that at the workshop, I have faith in our young people, is what I'm going to say, is at the workshop, our early career people were really very much into this being something that they wanted to see happen. And so that gives me hope that you know, there's a culture there that's potentially more collaborative than we have been in the past. The fact that so much of the community has to get involved in all of this also, I think, maybe tends to diffuse perhaps some of the issues we've had in the past. But you know, human nature is the way that human nature is, and so we may end up with some sort of these contentious types of things.
Now also,
but
just because people agree doesn't mean they're right.
No, that's absolutely true too. And I mean, we're not talking about everybody agreeing. We're talking about everybody going through the scientific process.
Yeah.
So I mean, there still may be, and in fact, you know, if you we actually did this in our 575 class, but you know, one way to get past sort of the cognitive bias towards a particular answer is to have kind of a team of rivals who work together on the same data set, you know, and they may have their cognitive biases, but at least they're in opposite directions. So yeah, I think there's sort of a misconception that we're all going to get together and it's going to be kumbaya, but it's really more all of us agreeing to a process and working through that, you know, a more rigorous, more robust look at these sorts of things, you know, as rapidly as possible, so that we don't have, you know, these debates hanging out and progressing only one annual conference at a time.
Yeah, so I have a questions about communicating the uncertainty. So in other sciences, we see that this is a real issue, like in climate science or in public health, when we are talking about the uncertainty of climate change studies or like vaccinations, effectiveness, we can see like the people on the other side using the uncertainty, like to recognize the uncertainties or like become confused on the topic. Do you envision something like that would happen in maybe the detections of biosignatures? Because maybe biosignature is not that intermediate ranges. If we are saying we are not detecting biosignature, maybe not a big deal. In the long run, it may hurt like funding or maybe something like what happened to the title signatures committee in 20 or 30 years ago. This NASA just cut down this bad.
Okay, so I missed the leap between having this communicable uncertainty to the funding cut. What was that? Yeah, so I'm thinking about like what the topic is lost inches, or I don't just become so confused about uncertainty. Right. They so just stop like maybe we should not do that. Yeah, so I think part of, okay, so try and redo the question. So you're pointing out that, you know, other fields like climate science and medicine, and especially, you know, at the moment with COVID-19. And by the way, we had representatives and people did in fact discuss some of the scales that had been used by those fields as well. So that was part of both our pre-work videos and as well as the discussion that went on there. So, you know, there are other examples of other fields that try to convey uncertainties to the public and have methods for doing so. But you argued that, you know, if you show that there's uncertainty, that potentially that means that people discount the field. Is that right? Yeah. And, you know, and the example then is, of course, SETI, when, you know, they did this search and, you know, didn't potentially find anything. And then, you know, SETI funding was essentially cut by Congress for quite some time. So I think that's exactly what we're trying to avoid here is this idea that it's either or, and if we don't find it, then it's failed. I think that's what NASA in particular would like to avoid. They would like people to see this as sort of a grand arc of measurements that need to be made successively by multiple missions and not just, you know, either or and therefore we failed. And so I think this is a way of inoculating our science community and the public to the fact that this is sort of a generational quest at some level to really get more and more definitive with time. So I think they're trying to avoid exactly that. But we did also talk about, you know, we just put together a framework in this. We had sort of grand plans that maybe we get to, oh, look, it's a confidence level 7, you know, what does that mean? But I think we need another workshop to really say, you know, how do we want to convey that uncertainty? Do we want it to be, you know, a color level rather than a quantified number? How do we actually do a quantifiable number? You know, and how do we assess the uncertainty at each level? So there's still a ton of work to be done in this area, but we kind of just took the first step into trying to understand all of that. Sorry, Mike. And then
So
we'll
data sharing and code sharing is something that a lot of communities have addressed. Are there real barriers in this community?
No, I don't. There have been in the past, I think. Sorry. So the question was that code sharing and data sharing is now just the norm. So are there any real barriers in the astrobiology community in that regard? And I would say that it's definitely gotten better than what we've seen in the past. And I won't call out specific things, but there were places where if data had been made more available, things could have proceeded faster in the past. But I agree that we are moving towards an open source. If anything, the workshop just validated that and said, this is good, we need this. You know, and is there anything we can do to enable or even make it even faster? Is there like a central repository where we can put things, or is there something we can do as a community to actually help that process? Megan.
So I was just wondering, I did see it touched as a field that could use like more discussion, but when actually quantifying uncertainties, how much was that flushed out? And also, is there a way for like, let's say you take one measurement and you have this level of uncertainty, and then down the line you take another measurement. Is there a way to then go back and have that sort of evolve the previous uncertainty based on new data?
Yeah, so the question was, you know, how much of a discussion do we actually have on quantifying uncertainty? And when did we also consider the fact that as we have more measurements, we may in fact become less certain? We could go backwards, for example, in our assessment of how certain something is. Yeah, we definitely discussed that. And we discussed the fact that there should be a double arrow on that sort of 1 to 2 and whatever, that in all cases, it could go in both cases. As you got more information, you know, you could actually become less certain about a particular hypothesis. So yeah, that was understood. The discussion of quantification, we did discuss it, but we didn't really, you know, in the time available to us, we did not coalesce around, you know, how we might actually do that. And I think that's like the top priority for the next workshop, is looking at how we get statistics and uncertainty actually into the framework. And we would have a more focused discussion with a different group of people that involves more statisticians, more data analysts than we actually had at this meeting. We did have some great statisticians at this meeting, but they were certainly a couple of them.
My question is kind of similar. So was there any discussion at the workshop about maybe going backwards in this process, or what that would look like? For example, I'm thinking of an example from my field where maybe you're very certain on questions 1 and 2 that the answer is yes, we have a signal and it's a real signal, and then later down the line you find out actually it was an artifact. So I guess, yeah, was there any discussion of what that would look like in the field, or is the idea that you wouldn't be allowed to move on until you for certain like ruled out that it can't be an artifact?
Yeah, no, I mean, there's no you wouldn't be allowed thing anywhere in here. I just want to point that out. But so the question is that, you know, again, just sort of echoing Megan's question, is the idea that, you know, that science doesn't flow in this linear fashion forward, and that potentially you can go backwards as well. And, you know, did we talk about whether or not people would not be allowed to go forward until they reach a certain level of confidence or whatever? I think, again, it's not the not allowed thing. It's just, you know, at what point do we really want to invest? You know, if you're an outside watcher at this, you might say, do I invest my science time and my career in trying to assess this biosignature if it hasn't reached this particular stage, right? So it's kind of risk management and sort of a statistical look at, you know, sure, you might be very confident about something, and built into the process in 1 and 2 is like having other teams verifying it, having multiple observations. You know, so at some point you're going to get to your comfort level where you're like, yeah, I really think this is real. I'm going to go after this kind of thing. But at the same time, the next step, it might be, oh, look at that. It's, you know, it's something that was in the instrument or whatever. And honestly, we just have to do our best as scientists. I mean, that's all we have to do. We know that we can make mistakes and that things might be as we appear, and that's the joy of science, is, you know, revealing that next piece of information that actually challenges everything you thought before. So yeah, and again, I just want to stress, this isn't driving towards, again, some kind of, you know, some kind of enforced consensus or anything like that. It's really about just, you know, enshrining the scientific process more than anything else, and just saying that, you know, these claims are so important and so impactful and of such high public interest that we really need to, you know, we really need to be very rigorous about how we look at them, and that we as a community are willing to help out with this process because it means a lot to us. I think everybody at this workshop was like, you know, this means a lot to me. So yeah, so we're just trying to work out kind of a framework for best practices, and just to help guide how we might look at these things. And of course, everything will evolve. We'll come up with some framework and then it will evolve over time. We expect this to be a completely living structure.
Well, I think that's a pretty good note to end on. So why don't we thank Vicki again for a fantastic question.
I was gonna